everybody, and welcome to our November webinar of the OCB Working Group BECS, which stands for Benthic Ecosystems and Carbon Synthesis. My name is Jessica Liu. I'm a research scientist at the NOAA Geophysical Fluid Dynamics Laboratory. The goal of our working group is to improve understanding of the benthic-pelagic interface and how processes at the seafloor influence ocean biogeochemical cycles. We host webinars on various topics, roughly bi-monthly. And just as a plug, if you are at all interested in giving a 20-minute webinar on your research, please fill out this Google form, which I will drop in the chat, or May will drop for me in the chat. Our working group applications have closed, but in advance of our working group meeting in April, we're soliciting feedback from the broader community on research priorities in benthic pelagic coupling, also via a Google form that we'll drop in the chat. We anticipate that if you were to fill out the full form, it will take 10 to 15 minutes, but we really encourage any answers to this questionnaire. So please fill out a subset of the questions if you find the whole thing to be too long. All right. Thank you, May. Today we have the pleasure of hearing from two speakers working on elemental cycling at the benthic pelagic interface from regional to global scales, Christopher Soames and Kanchan Maiti. First off, starting us off, Christopher Soames is a research scientist in biogeochemical modeling at GEOMAR in Kiel, Germany. He received his PhD in 2013 from Kiel University in the GEOMAR Helmholtz Center. He works on a broad range of topics, spanning using isotopes in marine sediments as a constraint for understanding ocean biogeochemical cycling in the past and present, and more recently modeling global scale iron and nitrogen fluxes using our system models. And so that's what he's going to be talking to us today. So Chris, do you want to go ahead and start sharing your screen? Yeah, take it away. Thank you so much.
Okay. All right, yeah, thanks. So it's great to present here. Yeah, so I'm Chris. I'm up here in northern Germany, and what I'm gonna try to make a case for today is that we need to think more about having a good, strong, robust, reductive, sedimentary iron release as part of our thinking of the iron cycle and also in our models. Right, and so yeah, before I move on, I also want to thank my collaborators here and colleagues in GEOMAR. They've contributed to some of these model formulations I'll be presenting, and also my thinking and view of the iron cycle and its sedimentary component. All right, so yeah, I'll quickly kind of go over what is the current state of the art in the global iron flux estimates, and also a little bit on how they're implemented in most of the global coupled three-dimensional models, right? And then talk about some of my simulations showing just how important a strong reductive sedimentary iron release can have on our simulations of marine biogeochemistry in the Anthropocene. All right, so just to quickly go over, a quick this is the basic view of the marine iron cycle from one of Al Tagliabue's papers, and so we think the main sources of iron to the ocean are from the margin sediments, also the more well-known dust, and also the hydrothermal vents. All right, and that, and these sources in the deep ocean then, of course, have to be upwelled, and then they supply the iron for productivity for things like phytoplankton, and also I'll talk a little bit about their importance on nitrogen fixation. Right, and just to quickly mention, I'll mention scavenging a little bit. So I'll kind of refer to that more generally here as, the many processes that essentially strip out the bioavailable dissolved iron into the particulate phase, and where it can sink back to the sediments, right? But the main focus of this talk is going to be on this flux right here. Okay, so I mean, so here is this another kind of review paper looking at the state-of-the-art global ocean biogeochemical models with the iron component. And we don't have to focus on all of these, but I just want to quickly point out what the range of, for example, the dust, which is kind of this more well-known input, and also the sediments, which actually has the largest uncertainty and also the largest values, although most of the values are, 100 gigamoles or less, but I mean, there is a very large uncertainty here, right? And so then if we just focus on a couple examples that has a similar input of dust, but very different inputs of sedimentary iron release going into the ocean. And what one might expect is that this model with a really high sedimentary iron release is going to have, a much higher levels of iron. But that is also not necessarily the case. It's because there's so much uncertainty in this scavenging, this process that strips out this bioavailable dissolved iron, that what most models essentially do is just increase their scavenging rates, no matter what their source inputs are, until they can just get kind of an average iron concentration, and more importantly, have like things like the Southern Ocean limiting iron as this most limiting nutrient. So, and what was more troubling is that these models had very little to go on, when they were first published. So, I mean, it was almost kind of guesswork before like the geotraces that really provided a good global view of the dissolved iron distribution. And the important thing here is that these models then with very different sources have very different residence times. So, I mean, this model that has a very small sedimentary iron input will have a iron cycle that turns over on multi-centennial time scales versus the one with high fluxes of sediment will have a subdecadal, which means that we also don't really know how sensitive the global iron cycle is towards climate change. And so what I'm going to do in a couple of slides, now that we have a much better observational data set, is to see if we can get some insight onto which at least spectrum of these rates are more realistic. All right, so but to first talk about, how exactly do these models calculate and predict these fluxes, right? And there are some models that still take this very simple approach that just essentially prescribe a flux from all seafloor grid points, right? And some can put something like a depth dependency to somehow account for higher rates in the surface ocean. But some models still just have a very simple approach here that's following just kind of a steady geologic source, essentially, to the ocean from the seafloor. But much more commonly is implementing a parameterization of a reductive sedimentary iron release. And for example, here's a couple examples that I'll kind of overview in the next couple of slides. And these are very pragmatic and easy to implement because they're based on a variable that global models already include, right? So we'll kind of review that. And I was really surprised. So I spent a little bit of time digging through the literature and I couldn't find a global coupled three-dimensional model that has a coupled sedimentary component with a dynamic iron. I mean, I found a couple examples that claim they did, but actually when I went through their references, I had trouble finding the actual description of the iron. And I know some groups are working on this, but it just kind of goes to show that, this this important flux has not, I think, really been addressed by the community like it should be. So now just to look at by far and away the most commonly implemented parameterization, and it dates back to this Elrod 2004 paper in GRL. And what we have here on the y-axis is this dissolved iron flux measured by benthic chambers. And on the x-axis is the carbon oxidation, right? So this is a reductive process that's where these mostly iron oxides are going to be reduced to this bioavailable dissolved iron 2, which can then flux out of the pore waters in this dissolved form. Right, and we can see in the filled symbols, we see this very nice relationship. And because global models already include this organic carbon flux into the sediments, this is something that's very convenient for us to kind of implement and see and quickly predict. Right, and yeah, but there's a couple symbols that really pop out, right? And that aren't actually included in this basic relationship. And these are all in very low bottom water oxygen conditions, right? And we know this is a anaerobic process. So when you essentially have less oxygen fluxing into the pore water sediment, you're going to switch to these anaerobic processes more efficiently, essentially, and have these higher fluxes, which actually is not included in this basic implementation of this in this LROD paper, right? So some of my colleagues here at GEOMAR, they kind of went through the literature and found even more data with a wider variety of bottom water oxygen where this dissolved iron flux has been measured. So again, we have this dissolved iron flux on the y-axis and carbon oxidation on the x-axis, and we can see kind of a similar relationship, although it's somewhat nonlinear here. But there's also a lot of spread, which is really interesting, can be mostly explained by this bottom water oxygen, right? So when we have these red and orange symbols, you can see these are all in very low bottom water oxygen, especially the red, right? And you can actually explain almost all this variability that you can't with the carbon oxidation rate with this bottom water oxygen. So what they then argue in this paper is for much higher rates, particularly in low bottom water oxygen conditions. And then they took their own benthic model and found that you could actually predict the dissolved iron flux actually very well using this very simple equation that has an equal dependence on the carbon oxidation rate and dissolved iron flux. And so what I'm going to do going forward now is essentially compare some simulations where I use this LROD formulation, which has a relatively low rate, and this Dale et al. Formulation, which has this high dissolved iron flux, particularly under low oxygen conditions, bottom water conditions, I should say. And so what model that I use is this biogeochemical component within the UVic Earth System climate model. It's a fairly coarse resolution, right? And it has a simple atmosphere. So we essentially force the ocean with these wind products. So because of these two things, it's typically classed in this Earth system model of intermediate complexity. And I just kind of have a cartoon of our ocean biogeochemical component. So we have phytoplankton that take up our nutrients, nitrate, phosphate, and dissolved iron. And also a diazotroph class, which can undergo nitrogen fixation, where also iron limitation plays a very strong role, right? I mean, we also have denitrification processes, which I won't talk about much today, but and again, what we're going to focus on is how we essentially calculate this flux from the sedimentary iron release. So we do have the scavenging process where dissolved iron is stripped out to particulate organic iron, and which sinks back to the seafloor and is modulated by a variable ligand parameterization. Okay, well, we also have the sources from the atmosphere. All right, and of course we have these full ecosystem interactions, which I'm not really going to introduce today. But again, we're going to focus on this flux here. Right, and as I kind of mentioned, so I'm going to have this base simulation with this LROD formulation, which has this very relatively low global rate of 15 gigamoles versus this Dale formulation, which ends up with a 114. So it's really substantially higher flux, right? And it also has this bottom water oxygen dependency. So I've also kind of tuned my scavenging. So each simulation has the equivalent global dissolved inventory, right? And so now let's redo this comparison with the dissolved iron observations to see if we can get some sense into at least which simulation is most realistic, right? And so here, let's just look at some snapshots of then dissolved iron in the upper 250 meters. We have the observations here, and then we have this base formulation with this relatively low rate of iron release. And so we can see that we're not really predicting and reproducing these high dissolved iron values we see on these continental shelves in particular. So by almost a factor of 2 in general. Right, and so work has already been published a couple of years ago, so I won't focus on it too much, but just to put some context here is that this simulation has a global dissolved residence time of 36 years. So now if we compare to this simulation with this much higher rates, right, we have kind of a similar, and then here is the difference between this rate and the LROD formulation, the base model, because we originally also used this LROD formulation, right? And as you might expect, where we have low oxygen, particularly in the low latitudes, we predict then much higher dissolved iron fluxes and the resulting dissolved iron concentrations, which now are starting to reproduce the observations, at least in this low latitude region, much better. And one thing that kind of jumps out is that we're still not reproducing the high latitude high values we see on the more broader high latitude continental shelves, right? And so, this, even though this rate is very high, it almost appears that it still could be an underestimate. And, there could be a number of reasons. Perhaps my coarse resolution model, I think, is probably not resolving in the vertical, is probably not resolving the low bottom water concentrations that can form just above the sediment interface. So that's probably one of the main reasons why I can't get this high flux there. But there are a lot of uncertainties all the way around. But just to show that, then this formulation now has iron cycle that operates on these sub-decadal timescales, which mean it can be very sensitive to environmental perturbations, including kind of in the Anthropocene, especially. Right, and that's kind of the exciting thing here. Now let's test how these two model formulations then respond in just kind of a very simplified warming scenario where I just prescribe increasing atmospheric CO2. So we get about 2 degrees of sea surface temperature warming on average. And I'm also going to have one other anthropogenic forcing that includes these increasing nutrient deposition from pollutants, mainly the result of different combustion processes, but for nitrogen and also iron, to see how this can also stimulate productivity and also some of the sedimentary iron release, for example, and then see how some of these feedbacks may play out in the model, right? And so if you weight it by the total N input, including nitrogen fixation, this will then predict about an 18% of increase of nitrogen to the surface. And for the iron, these are all from independent atmospheric chemistry models that I just forced the model with. And then from this Biogeochemistry paper, they estimate about a 24% increase. Right, and so this is again from year 1850 to 2100, kind of this typical Anthropocene scenario. Right, we just do these kind of idealized simulations. Right, so let's first look at productivity. Right, and so as we saw, the main importance was in the tropical ocean. So I'm just going to show some averages looking at the tropical ocean, right? And so this is net primary production. And so the y-axis is going to be the change relative to pre-industrial. And then I'm going to just kind of zoom in on this period from 1900 to 250. And what happens in most models is that the warming ends up stratifying our ocean and reducing the input of nutrient-rich deep water to the surface ocean, which results in a slight decrease in NPP, especially in the low latitudes, right? And so that's what we can kind of see happens here in this base simulation, right? And then if we also look at the volume of oxygen deficient zones, when we have less productivity, we have less oxygen consumption at depth. So we also have a slight decrease in our oxygen deficient zone volume, which is inconsistent with the data. I'll show a little bit later, right? But what's interesting is now when I include this stronger reductive sedimentary iron release, the system actually flips now to an increase in MPP and now expanding oxygen deficient zones over this time period. Right, and so it's already showing that this one arrow, just slightly changing this one arrow, can already have an important influence on how tropical ocean biogeochemistry may respond in the Anthropocene, right? And then if we also continue to add more nutrients to the surface from these deposition estimates, we also can get even higher NPP and even stronger. So the nutrient pollutants may also play an important role on its productivity and expanding ODZs. All right, and of course, so to show what is then the sedimentary iron release doing. So in this base formulation, right, it's only responding to essentially the carbon, the particulate organic carbon that's sinking and then oxidizing in the sediments, right? And and since NPP decreased in this scenario, we also have a slight decrease in the base. Whereas in the sedimentary high simulation, we also have lower oxygen conditions prevailing, which really increases the sedimentary iron release according to this formulation, right? And so now again, we kind of flips the system from having a decrease in sedimentary iron release to an increase. Right, and it was also interesting that it's actually quite sensitive to these, also the deposition input, because a lot of these low oxygen areas are strongly nitrogen limited due to denitrification. So especially when we added nitrogen into these systems, that actually stimulated more productivity, which then drove even higher sedimentary iron release. Right, and I do have to quickly mention nitrogen fixation, because this kind of feedback doesn't really operate without it, right? So if we look at this base simulation, we just have a slight increase in nitrogen fixation, so not much of a change. But on the other hand, when we also add this extra iron, this is essentially the main limiting nutrient for them in much of the low latitude ocean. So this actually stimulates a really strong input of nitrogen fixation. So when we're getting this extra iron release, not only are we getting extra iron, but then we're also getting this extra nitrogen in the system through nitrogen fixation, right? So it kind of developed this amplifying feedback between sedimentary iron release, nitrogen fixation, where it then further enhances NPP and the oxygen deficient zone expansion in the model, right? And I can also show nitrogen fixation wasn't as sensitive to the deposition, which is not really important here. But now let's to kind of start to wrap up a little bit, let's look at this deoxygenation trend. So up here are the observations of the loss of oxygen in the upper ocean. And models in IPCC models, these global coupled three-dimensional models really struggle to reproduce the deoxygenation, especially in these low latitude oceans, right? And so we can see if I just take away all these biogeochemical feedbacks, my model actually will predict an increase in oxygen, which is inconsistent with the observations. Whereas if I include all of these biogeochemical feedbacks that can amplify productivity, we can see now it's starting to flip this system to increasing NPP and increasing oxygen deficient zones, increasing deoxygenation, we can say. And so if I kind of zoom in here, so we can actually see the difference a little better, also on the scale here, we can see that this biogeochemical feedback between sedimentary iron release and also nitrogen fixation exactly operates in these low latitude ocean, where many models are struggling to reproduce the deoxygenation. So it could be an important missing piece to tropical ocean biogeochemistry that's to some degree fueled by a strong sedimentary iron release, right? Yeah, and just to kind of sum up what I described here is that, when you have this increase in NPP, you can get more flux to the seafloor, lower bottom water oxygen concentrations, and additional iron release, which can potentially create a positive feedback on productivity. But the problem is you also have strong nitrogen loss and nitrogen limitation that you have to break to essentially get to higher productivity levels. So what happened in my model is this iron release actually stimulated nitrogen fixation, which kind of could break this strong negative feedback from the nitrogen cycle side, and also then result in actually an expanding oxygen deficient zone and deoxygenation, which the observations are showing. So, and most models struggle to reproduce. So I'll just kind of sum up there. So yeah, I've kind of talked about these two main points, and I also want to say some kind of exciting news going forward is I'm going to continue this work. And so I do have a new project to actually not only look at the reductive sedimentary release, but another really aspect that's missing is the non-reductive. I don't know of any global model that's actually included explicitly a non-reductive source. So that's kind of what I'll be working on with a new student that actually officially starts next week. And we also want to put iron isotopes in the model to help constrain these processes. So kind of that's kind of where I'm thinking moving forward, and I'll wrap up there. And yeah, I don't know if I take questions now or later, but I'll hand it over. Thanks.
Thank you so much. I will take questions now. So you're welcome to raise your hand or put questions in the chat for Chris. Actually, I'll start us off. I was wondering about the phosphorus cycle. I'm not super familiar with MOBI, but I was wondering if you see, I mean, are you representing the phosphorus cycle, and do you then see a limitation to the climate change effect due to essentially phosphorus limitation that you would get in the global oceans?
Well, I mean, we have phosphate in the model, but it's at a constant inventory. So we don't have the sedimentary fluxes for the phosphate cycle, right? And, I mean, well, the phosphate, we think it operates on much slower timescales compared to the iron. So I think, so it depends on kind of the timescale you're thinking about.
Yeah, okay.
But I mean, essentially, when you compare nitrogen limitation to phosphorus, you do see this slow switch towards phosphorus limitation because there isn't a process like nitrogen fixation that can, relieve phosphorus limitation at the surface ocean.
All right, thank you. Will Carlson.
Yeah, hi. Still loading my virtual background. Can you hear me okay?
Yes.
Yeah, hi, Chris. That's a very nice. Sorry for the video not clicking on quite yet, but the complexity of the benthic flux, obviously, I mean, it's obvious to many of us that there's not a single parameter, and there's probably not two parameters that can easily describe why you sometimes get high and low iron flux. A lot of it has to do with little micro environments that are awfully sensitive to the kind of benthic biota that are present in any given system. I think we can all understand that. What do you think modeling is going to move towards greater and greater complexity in trying to, actually arrive at process-driven benthic fluxes, or do you think we're really at the limit of that and we got to keep it simple with just a couple of parameters as you've shown?
Well, I think we need a variety, right? So, I mean, is important. You also see these benthic hotspots, I know. And, so I think that's where we kind of have to learn from high resolution models that can perhaps even at some point try to resolve these benthic hotspots, right? And I think then you have to kind of work from that towards a more larger scale into something we can really implement into the global models. Right. And so, I think you really have to start from these small scale models and derive some relationship and how many hotspots are on average are in these certain regions and why, develop some type of relationship from that we can then apply to these global models because. Right. And I think that's the way forward. I mean, there's no one perfect model, and I think it's good to have these more complex models, but which have their scale limitations due to computational efficiency, and try to scale these up to I mean, even really high resolution models, because one limitation to all the results I show here is we don't resolve many of the narrow continental shelves. So in principle, I should be underestimating all these fluxes to a large degree, although these parameterizations can already predict them to be quite high. So I think, just a variety of models and try to learn from it and parameterize them into global models is, the approach to take.
Yep, got it. Thanks.
We have a comment in the chat from Anfam. Do you want to unmute or do you want me to read it?
Hello. Yeah, it's just a comment that There is a model by Begura et al. 2019 that they will try to include the inorganic particle of iron released from sediments in their global model, and I think which might reflect the non-reductive source of iron. Just one comment. Thank you.
Okay, so all right, yeah, okay, yeah, okay, thanks. Yeah, if you could maybe even write that in the chat so I can check it out. I mean, in case I missed it, that would be great because I could think. I'm not really sure.
Yeah, I paste it in the chat.
Thanks.
All right. If we don't have any other questions right now, then let's move on to our second speaker. And at the end, if there's any other questions for Chris, you can pop them in the chat. All right, so I'm happy to introduce Kanchan Maiti, who is a professor and chair of the Department of Oceanography and Coastal Sciences at Louisiana State University. His research group focuses on the transformation of carbon, nutrients, and contaminants in the coastal and deep ocean systems. His work crosses the land to ocean aquatic continuum and focuses on processes spanning the formation of hypoxia, ocean acidification, extreme events, looking at benthic fluxes with influence to biological pump and air-sea fluxes. So yeah, Kanchan, why don't you take it away?
Thanks, Jessica. Thank you all for joining at the webinar. So today I'm now going to kind of bring it to a more local scale from the global scale that Chris just talked about. So I'm mostly going to focus on the Northern Gulf of Mexico, Mississippi River influence shelf. And what I'm going to talk about these two projects funded by NSF Chemical Oceanography. One to look at how benthic processes impact carbonate chemistry in this shelf area influenced by Mississippi River. And the other offshoot of that project that got funded after Hurricane Harvey is to look at how does tropical storm impact the carbon budget in this area. So I kind of combine them together to give a synopsis. And I want to start off by acknowledging my PIs and collaborators, Woo Jung Kai and John Lerreter, who are part of this project. And also the students, Hannah Dye. This is a lot of this work I'm presenting is her dissertation. And Okil and Byron, who just went on these cruises, although it wasn't part of their project, and helped us a lot. So with that, before I start talking about my backyard, which is the Mississippi River Delta, I just wanted to kind of widen the focus and talk about all these river-influenced deltaic systems out there. And wanted to point out that when we talk about this river-influenced continental shelf, we are talking about huge areas it has influence on. About thousands of kilometers of the continental shelf are influenced by these rivers, where sediments and organic carbon fueled by the productivity in the water column gets impacted. And there's this cross interaction between biology, storm, wind, currents, all of which comes together to impact both the bottom and the water column chemistry. And when we start looking at the system globally, we realize that these continental shelves, especially the river-influenced continental margins, they account for over 40% of the carbon burial in the global ocean. So these are really huge areas with a lot of carbon, and there's a lot of benthic processes that is going on here that is interacting with the shallow water column above it. So what goes on in this system? So if you think of a typical continental shelf where there's a river coming in, you end up with a weak stratification or a strong stratification depending on wind and river discharge in the top layer. And if we look at some of the recent papers, most of them suggest that most of these river influence system used to be net source of CO2 pre-industrial age. Now, with more nutrient in these systems, they act as either sink or weak sink of CO2. So that means there's a lot of productivity that is going on, and a lot of that carbon is either sinking to the bottom in the shallow shelf or moving outward. And that's where a lot of these uncertainty is there. What happens to this carbon after they're produced? Are they all sinking and there's a huge benthic flux, or are they just moving out? And with that in mind, I kind of started this project, and we're looking at this shelf area right here. This is the Mississippi River. This is the hypoxia zone. There is a typical hypoxia that happens every July, August in this area, where the oxygen concentration goes below 2 milligram per liter in the bottom water. So suggesting there's a lot of organic matter mineralization that is going on in here. And on the right, I have these stations. So there are quite a few stations, but today we'll focus on these 4 stations, B5, C6, D3, D5, because these are the repeat station for 2 years. And the idea is, what is going on when this organic matter is getting remineralized? Are they releasing DIC and or alkalinity that might impact the bottom water pH, and how that impact the carbon chemistry in this area? And then the next part of it is what happens when there is a episodic events like storm, and how does that disrupt this carbon and oxygen budget? So the main tool for this work is, oh, sorry, I skipped a slide here. So before we go into the actual data, I just wanted to kind of introduce the uncertainty in this system in the Northern Gulf of Mexico. And this is actually a really nice summary of the carbon budget in this Northern Gulf of Mexico. It came out of one of the OCB workshops. And the most interesting thing I wanted to show here are the number of question marks are more than the numbers that we see in this. And the 2 that stands out, one in blue, is how much of that carbon is flowing laterally. We have a little bit better control on that based on some of the work that has been done in recent times. And the other one is, what is the benthic exchange in this shallow shelf with the overlying water column? And that's still an outstanding question, and something that we should look at in greater detail. So now going back into our actual research and the tools that we use for this work. So we're kind of using, starting with the use of just oxygen. Oxygen is simple, easy to use, to understand it as a proxy for organic carbon mineralization. And then we'll move on to other carbonate parameters. But most of this work was done in situ using the Benthic lander system shown here, right here. It basically has 4 chambers, and when it goes to the sea floor, it kind of traps about cubic feet of water with the underlying sea floor, and this acts as a closed system. So when you have change in concentration in these boxes, that has to be either something going into the box from the seafloor or going out of the box into the sediments. And that change in concentration over time allows us to calculate the flux. So what it gives us an idea about the total flux. And in this case, we're looking at oxygen. So this kind of tells us what is the total amount of oxygen that is used up in this sediment. And that we can represent as TOU for this talk. And so TOU represents sum of all oxygen processes. It could be benthic processes like biological processes, microbial processes, diffusive processes. And then we have a microprofiler system that goes right next to this lander and sits on the sea floor and slowly penetrates the sediment at very high resolution. We're talking about 250 micron resolution for this work. And it allows us this really nice gradient of oxygen as it penetrates the sediment. And we can use this profile of oxygen and use Fick's law to calculate just the diffusive part of the oxygen consumption. So now we have 2 in situ method. One gives us the total oxygen consumption, TOU. The other gives us DOU, just the diffusive component. In an ideal world with no biology, no other processes, TOU and DOU should be equal. And what most people have seen, including this work, it is usually not. And especially in the shelf areas, this difference between TOU and DOU, with TOU being more than DOU in most cases, this represents the organic matter that is being used up by the benthic fauna and bioirrigation and all these biological processes. So there, like a little bit going on a tangent there, we can actually understand even the impact of biology from just this oxygen data. And that is really important, as we'll see here in the next few slides. So we did this 2 years for 4 stations. And these are the 2 years. We have the dissolved oxygen for this area on the left, and salinity, dissolved oxygen on the left and salinity on the right here. One thing that stands out, 2018 had much stronger hypoxia. The red area here is oxygen concentration, which is less than 2. And also, not surprising, this area has lower salinity the same year because there was more river influence. It was not the case in 2019. 2019 was very different, and the salinity impact, as you can see, is much saline. So the river impact is less, but most of it is likely due to the passage of a storm. So in 2019, we actually sampled a few days, about 5 days after a Category 1 storm moved through the area, so which made it a little bit more dynamic system. Although we really wanted a perfect comparison, there's nothing like that in oceanography, so we have what we have. So those are the 2 years, although the same station, the physical processes are probably different. So I'm not sure how good a comparison they will make, but nevertheless, they just do give us 2 years worth of data to look at. And what we can see here in this figure on our right, blue is our TOU, our total oxygen consumption, and orange is our DOU. 2 years, 2018 for those stations, and repeat stations again in 2019. The numbers in gray, those represent the oxygen penetration into the sediment, 2 millimeter, 2.5 millimeter, and so on. But the main thing here, which was actually quite surprising for me, is TOU is a lot higher than DOU. So we got into this work thinking that this is a hypoxic area, very low oxygen in the bottom water, probably very little benthic fauna in that area. But it turns out that's not the case. This data suggests, and I found it actually contrary to what we hypothesized, that there is a big difference that we can attribute to benthic fauna. So we looked up some of the data out there. There are lots of polychaetes in this water that can live at really low oxygen concentration, and we think that is what is causing this difference between TOU and DOU. But still, I thought it was really interesting to see that much benthic contribution in this low oxygen environment. So we went and looked into some other data from other areas. We have the Rhone Delta data and Congo Delta. And to my surprise, I found the difference between TOU, or the amount that we attribute to benthic fauna, is actually quite high for Mississippi River shelf compared to Rhone or Congo. And I think that has to do with something. For Congo, we are talking about really deep water, so that makes sense that TOU and DOU are closer to each other. And the other interesting thing about Rhone, I thought it would be more closer like Rhone, but it is not. But Rhone Delta also have very high sedimentation rate. Maybe these fauna are getting buried, which is causing the DOU and TOU to be similar. But what is really interesting and what is something to keep in mind is that these benthic fauna do impact the carbon cycle in this area more than we probably think it should. So that's kind of overview of the oxygen consumption. We can think of TOU now as kind of, if we use the rate fill ratio, that kind of allows us to estimate what is the aerobic respiration that is going on, how much DIC will be produced from TOU. So that gives us an idea of inorganic carbon produced from respiration through aerobic respiration. But that doesn't tell us the whole story, because there's a whole lot of anaerobic respiration that is going on in the sediment. So in that case, we can kind of start comparing TOU with DIC fluxes. So my next slide kind of shows that. So now, same data set, same stations, we have in gray DIC fluxes, which kind of represents both aerobic and certain anaerobic respirations and the fluxes of DIC out of the sediments. Then we have TOU, which is mostly representative of aerobic respiration, and we have DOU too. But what is interesting to note here is the DIC fluxes obviously are much, much higher than TOU. Sometimes the oxygen fluxes only represent 10% of the total DIC fluxes. So by the way, all this TOU is now converted to oxygen to carbon fluxes using rate fill ratio. So when we look at this, with the DIC being so much higher than TOU, we start thinking there must be some alkalinity fluxes that is going on, because we know anaerobic respiration also leads to production of alkalinity. So the new figure that now I have on the other side kind of shows the DIC and the alkalinity fluxes paired for each of these stations. And the data is all over the place. As we can see, it looks like there's not always a positive alkalinity flux for each of these stations. In fact, some of these stations almost act as a sink for alkalinity, while other few stations do have significant alkalinity fluxes, like we can look at D3, B5, and D5, where we have positive alkalinity fluxes, whereas other stations, there is almost no alkalinity fluxes or even a negative alkalinity flux. So what we looked at now is like, okay, if we look at this data and start thinking about some of these stations where we have positive alkalinity fluxes, what is special about them? And one thing that kind of strikes out is when you look at the ratio of what percent of the total DIC flux can be represented by oxygen or aerobic respiration, that's the TOU to DIC ratio shown here as percent on the secondary axis. And we can see for D3, B5, and D5, the blue dots represent those values, we have a lot smaller fraction of total DIC represented by aerobic respiration, which kind of starts to make sense. We can kind of say that, okay, in these cases, probably there is a lot of alkalinity coming out due to anaerobic respiration that is happening in these sediments, given that the aerobic respiration actually represents a smaller fraction. But nevertheless, this was also kind of surprising, and we're still looking at this data and trying to make sense of this, is the benthic alkalinity fluxes was very variable, quite variable across the 2 years across these stations. So what we did is we also took this alkalinity and DIC flux data and tried to compare them with some pore water data we have from these stations that was collected at the same time. And so now in this figure, we have the same DIC and alkalinity fluxes, but now we have pore water alkalinity to DIC ratio. So it's the average pore water concentration dividing the TA to DIC, with the yellow dots showing that ratio on the secondary axis. And what we see is that for the stations where we have positive fluxes, at least for the 2 stations where we have positive fluxes, the TA to DIC was greater than 1, or in other words, there was a source of alkalinity more than DIC in this pore water that might be fluxing out. But what left us scratching our head is it quite doesn't follow that rationale, which is for D3 it works, for D5 it works, where we have a TA to DIC ratio greater than 1. But for B5, where we have a positive flux, but we do not have a total alkalinity to DIC ratio in pore water, which is greater than 1, which was surprising. And it took us some time to figure out what is going on. And this kind of touches on what I'm going to talk about a little bit later, is how dynamic these systems are. So what we did, we went back and looked at our bottom oxygen concentration throughout the time the benthic lander was sitting at the bottom. And you can see for B5, shown here in orange, the system, it has really low oxygen, but somewhere around 12-hour period of our observation, the oxygen concentration went up. And we think what happened, and we collected our sediment core somewhere here. What we think happened is that we are looking at 2 different things, probably because of this mixing event, or maybe current flowing over the over this area with enriched oxygen water column. It probably flushed the pore water, this top few centimeter of pore water, with the bottom water, and the bottom water has a TA to DIC ratio less than 1, and that's the best rationale we have right now to explain this. But it is interesting, but it does allude to the fact that it's very hard. I think Will Barrelson was saying this. It's very hard to kind of point a finger to any particular thing to explain a lot of this data. But the take-home message from this is what we're seeing here is a lot of these alkalinity fluxes are not positive here, as we would have expected. And what we think is happening also is that a lot of these alkalinity that is generated by iron reduction is being lost, because we also had H2S sensors in this microprofiler system, and we did not find any H2S in the top 10 centimeter. And we know there's quite a bit of sulfur reduction that goes on in these sediments. So that kind of points to the fact that maybe we're removing a lot of these metals as metal sulfides, and that can lead to net alkalinity removal in this system. So the bottom line is it's a really complex system, and just using one parameter to kind of extrapolate it for the whole basin or whole shelf is not likely to work, because of all these interactive processes that can determine the DIC and alkalinity fluxes. And depending on the ratio of DIC and alkalinity, it will impact the bottom water in this area. And in fact, we also have some of these pH profiles in here. And I'm just going to point to this one because it has this surface pH minima at 15 millimeter, which often people have explained as alkalinity removal due to oxidation of metals. So there is that hint, but we haven't really looked at the data closely. So there is definitely a lot of alkalinity removal that is going on that might explain some of this data here. And just to kind of compare them with the bottom water, so this is kind of our benthic fluxes, but the end goal for us is how does it impact the bottom water? So what I have here is the bottom water profile for each of these four stations in different colors, 2018, 2019. I'm going to kind of get rid of dissolved oxygen, and because the master controller here is DIC and alkalinity. And if we start looking at this data, the bars here represents the sea floor. So the bottom sample is taken 2 meters from the sea floor in each case. And we can start seeing, at least for DIC, that DIC flux that we consistently see across every station definitely impacts many of these stations, because we see an increase or an enrichment of DIC in the bottom water. For alkalinity, it actually reflects pretty well. For example, when we look at DI5 in 2019 and B5 in 2019, we see a big increase in bottom water alkalinity. And these are also the stations where we see B5, D5, where we see a positive flux in alkalinity. So there is definitely reflection of these bottom water benthic processes into the overlying water column. And that is definitely driving the lower pH that we see in the bottom water in this area. But it is important to keep in mind that these are all an interaction of both aerobic and anaerobic processes, and we still quite haven't been able to figure out all the different processes impacting this. So with that, I'm kind of summarize this first half of the talk by saying that the bottom water chemistry definitely gets influenced by these benthic fluxes, and the interplay between DIC and alkalinity fluxes is driving the bottom water pH. And in most cases, we see a drop in pH in bottom water. And given that we expect a lot of changes in this area over the next 50 years or so due to river influence, discharge going up, and increased intensity of the storm, likely this area is going to see more drop in pH from benthic fluxes during hypoxia. So that's kind of my overview of this Louisiana shelf benthic chemistry and how it impacts bottom water under what I call usual conditions. So now I'm going to kind of switch gear to another interesting aspect of this study, which is what happens to this system, which is already we saw is a pretty dynamic system during storm events. So for that, we're going to go back during Hurricane Harvey in 2017. And that happened in August, and we're able to get a rapid funding and go out on September 20th, less than 1 month after Harvey happened. And what we didn't expect, and we probably didn't want, was we got into a category 1 hurricane when we went back out again in October to look at the impact of Harvey. And we actually got our own category 1 hurricane. Probably nobody have heard of NET other than people in Louisiana, but it was it was pretty scary out there. It was about 15 feet of seas, and we left the area, but we came back within 2 days of the passage of storm. So that gave us almost two perspective of impact of hurricane in this study. And just to give you an idea, we looked at how much sediments were deposited after these hurricane events, especially after Harvey. And we used beryllium-7 to look at the data. We can see there are these massive deposits of sediment using presence of beryllium and static and X-ray, of course. And these dots here kind of represent that sedimentation rate. And we can see a lot of these areas had new or redeposited sediments of up to 10 centimeter within a matter of few days. And just to give a perspective of average sedimentation in this area is in the order of 0.5 centimeter or less. So that's a huge amount of sediment that probably got remobilized and got redeposited across the shelf. And if we start thinking about this, just a back of the envelope, if we just be very conservative and think, okay, let's say there was a 4 centimeter layer of sediment that got deposited across this shelf up to 50-meter isobath. If you look at the carbon content of this sediment at about 0.5%, which is the usual value, we are talking about huge amount of sediment and huge amount of organic carbon that got injected into the water column. And just to give us a reference, we are talking about more sediment that got resuspended and deposited during this one hurricane event than the entire sediment brought by Mississippi River in a year. And all the carbon associated.
Wrap up, please.
Okay.
Thank you.
So, and what we see is that what it led to is a huge increase in DIC fluxes during this period. So it basically enhanced respiration in the sediments, and that was combined with also increased in respiration in the water column, mostly because there was so much resuspension, the productivity probably got reduced and respiration took off. And that led to this really interesting picture here. We see that green is before storm, yellow is after storm. We see that big jump in CO2 in the water column and a big drop in pH associated with it, which makes sense. Almost 0.1 unit drop, which is what we see on a global scale for last 100 years. And then when we compare with some previous data, we see that the system, which is usually at equilibrium or a weak sink of CO2, turned into a net source of CO2 after the passage of storm. So I leave you with the summary here. Main conclusion here is the tropical storms are an integral part of the system, and we should look at their impact on carbon budget and constrain them in for the bigger picture. And the other non-science conclusion I should say is never send a first-year graduate student on a cruise that involves hurricane. So that was the only student I've actually dropped out of my program after coming back from this trip. And that's actually me right there. Thank you.
Thank you so much, Kanchan. We are at our hour. If anyone wants to stay and ask a quick question, we can take that. I can, yeah, I see a question. I'll just read it. Thanks for this is from Jesus Campare. Sorry if I'm mispronouncing your name. Thinking about tropical storms can mobilize sediments. Do you believe that your differences in TOU minus DOU ratios with other estuaries could be related to the resuspension of rests from the oil spill of 2010, or even current drilling activities that affected your study area and could alter the local communities, such as epi to infaunal organisms? I don't know if hydrocarbons are commonly found in your samples.
Yeah, that's an interesting question. There have been a lot of PAH work from this area that has been done. In general, the PAH in these sediments are actually quite low because these are really hot systems. The temperature is really high. So most of the oil got remineralized very quickly within few years. So I do not think after so many years, it is an impact of oil and gas related oil spill. But that said, there are hundreds of oil rigs out there, and it's very hard to say if there are some leaks that we are missing. So cannot really conclusively say it's not.
Okay, and I see a hand raised from Christophe Raboud.
Sorry. Hi, Kanchan, I can see you, but yeah, okay. Yeah, well, I have a few questions regarding the importance of the anoxic diagenesis in these sediments. You know that we have published a paper where we basically showed that the sulfate concentration hardly decreased in these sediments. So it's hard to believe that if sulfate is not decreasing, there is a big wealth of anoxic diagenesis, except maybe the iron and manganese reduction in that system. So what do you think, how do you think that can be reconciled with the message you had on the alkalinity influx to the sediment, and how is it related to that lack of sulfate reduction somehow?
I don't think there is a lack of sulfate reduction. I think there is sulfate reduction for sure, because there's limited data that we have shows the sulfate reduction is about 10 to 40 millimole, right? So it is. So we haven't completely looked at the picture here, but a lot of it is actually, I think there's a lot of iron. I think the iron respiration is really high in this area. So we are removing a lot of that H2S as iron sulfide or manganese sulfide, as your paper showed, because iron and manganese reduction is really high in this area. So I think that is the only way we can think of reconciling that difference right now, because I cannot, the only other thing that is left out is calcium carbonate precipitation, which I doubt is happening. So that's what we are at. I mean, we still need to think about it, but maybe I'll chat with you offline. But yeah, it is interesting data, because I know Will Barrelson has a paper which showed mostly positive fluxes of alkalinity in 2011, not negative. But this one is slightly different. So we're still kind of racking our brain on that. Okay. Do you have any thoughts?
Well, you know, I think it's sometimes very difficult to estimate the fluxes of alkalinity. It would be interesting to go into the raw data to really look, you know, make a certain the value on the sign of the flux, because sometimes it's very limited fluxes, although these ones were pretty big. So it should be something you see very well. But as you said, it's a very dynamic system. So there might also be some, you know, leakages if the sediment is a bit sandy. So there could be also exchanges from the, so we've seen that before. So yeah, we can continue talking about that offline maybe.
Okay, thanks.
All right. Just one last question from in the chat, asking from Ryan Ahmed. I'm sorry if I mispronounce your name. Just wanted to know if there are any lasting effects on carbon cycles after the extreme events passed.
Yeah, that's a really, really good question. I do not have an answer for that, because we stopped at October, because we thought September 1 month after the hurricane, then October 2 months after the hurricane is good, but it got complicated by another hurricane. So it's kind of hard to say that, but my guess is it would have a longer lasting, given that the residence time of CO2 and oxygen are very different. One is much faster. Oxygen has a much shorter residence time compared to CO2. So I think the CO2 signature will be there longer than oxygen. Even if the oxygen gets reset, the CO2 may not.
All right. Thank you very much. Just a note that OCB also has another, I think, working group or workshop called Seesaw, focusing on extreme events. That if anyone is interested, link is in the chat. And so with that, I'll thank our two speakers today. Thank you again, Christopher Soames and Kanchan Maiti. It was a pleasure hearing from both of you.
Thanks.
Thanks.